Hello, today we're going to be talking about populations. And since we're just starting to talk about this, we're going to talk about how you measure population size in the first place, and when you look at population growth, some things that you should keep in mind. So this is the start of our fourth unit on populations and toxicology, and these are the chapters that we're going to be talking about in this unit. So if at any point you need to refer to something in the book, this is where you need to go. And basically, you can see that we're going to focus a lot on human populations. But before we get there, we need to talk about populations in general, and this does apply to all species, not just humans. So we need to really make sure we understand what a population is. So hopefully you remember from last unit that populations are a group of members of the same species that are all living together in one area at one particular point in time. So I'll give you a few examples of some populations. These are all populations. Now, not all of them specify the time that we're referring to. So a lot of times that happens, and if they don't give you a particular time or date that you're talking about, then you just assume that you're referring to the current population at this point in time. But sometimes you want to look at historic populations, and for those you will always have a date next to them. But I want you to notice that all of these are specific species and only one species in a specific spot at a specific time. And basically they can all also come in contact with each other. Now, all of these things are not considered populations. Most of these are actually communities. So remember, communities are what you get when you put a bunch of different species together in the same area. So most of these, like weeds, birds, fish, that's not a species. It's a larger group. And the other thing about these is not all of them specify a very specific place, or in some cases it might be too specific, and the range of these organisms in that area might actually be larger than what they say. So when we look at a population, we want to find out some things about it. And usually, when population ecologists look at this, first of all, they want to know, okay, how many individuals do we have in this population? But also, how are those individuals distributed? So it's important to look at how spread out they are. And that's something called population density. So we don't want to just know how many individuals in the population. We need to know how much space they take up too. So we need to know the population per unit area. And that's the population density, basically how spread out or how close together are these organisms. And that relates to the organism's range. If they need a bigger range or territory, then they need to be more spread out. So after we figure that out, we want to look at, okay, how does this population vary at different times? Is it growing? Is it decreasing in size? And then what's causing that? So what percentage of the population is dying? That's called the mortality rate. Or what percentage is born? The natality rate every year. All right, and these are all going to obviously impact population size. So when we measure populations, or communities for that matter, you try to do it by random sampling. Basically, we want to determine the population of the whole group of individuals based on just how many we find in a small area. And obviously, the larger your sample, or the more samples you get, the more reliable your data is going to be. Since you are randomly sampling, you're not going to get every single organism. So you want to include as many as possible. So there's several different methods to do this. One is to take a census. In a census, they're attempting to literally count every single individual. Obviously, this is really, really difficult to do. And if you've known anything about the national census, you can probably tell how much work would go into doing such a thing. And even then, you're not going to get every single person to participate in a national census, so you're probably going to miss some people. So a true census is very difficult. Another method with something like birds or anything that makes some sort of sound, you can basically count them based on how many times they're heard calling. And in individuals such as whales, they actually may have a specific voice for a certain type of animals. So that's one way. Another way is a habitat survey. You look at the environmental effects of organisms, and that can help you tell how many there are there. So they use this very often for beavers in particular. They can tell from the amount of activity that the beavers have had and the amount of chewing on the trees in the area. They can tell about how many beavers are there. Another method, very popular method, is collecting, trying to collect all of the organisms in a certain area, bring them back to a lab, identify them. We'll be doing some of that later on when we get into water quality. We'll be collecting some macroinvertebrates from the stream. Some sort of similar to that is a mark and recapture method. You capture the animal, you mark it, you go back out a certain amount of time later and you recapture it, or you recapture a group of animals. You see how many of them are marked animals, and that should give you some idea of population size. So that's called the Lincoln Index, and you can pause here for a second and look at this example. This is basically showing you how you would use that mark and recapture method and the Lincoln Index to calculate the population size. Another method is called a quadrant, and it's a quadrant because you usually do it in 4 square meter areas at a time. You mark them off, you count everything in the area, and then you multiply what you get, the average of what you get, by the total square meters of the habitat. So you're just taking a small area and you're assuming the rest of it is like that. Another very similar technique is called a transect, and basically you run a straight line, usually just a tape measure, down a middle of an ecosystem, and every, say, 10 meters or so along the line, you stop and look at a 1 meter square area and count everything in that area. Now, both this and quadrants are a little bit more accurate at figuring out biodiversity of a community rather than looking at population size. Now, there's a lot of problems when we're trying to count populations. First of all, a lot of these organisms are hard to catch. All of these methods take time and take money, and it has to be worth it to do it. The method of counting is really dependent on what kind of species you have and how you're going to find them. And the population is always in flux. It's always changing. So sometimes you really want to look more at what are those changes rather than how many individuals you actually have. So there's a number of factors that affect population size, and they're what you probably think of. So birth rate, remember we said that is natality rate, death rate or mortality rate, immigration, and emigration. So immigration is organisms coming into an area. Emigration is the opposite. They're leaving. So think E for exit. They're exiting the area. I for immigration. They're coming in. So if you want to look at the change in the population, you basically take your births plus your immigration. That's how many individuals you've added to the population, and then you subtract the sum of the deaths plus the emigration, how many individuals have been lost. So this is just another way to show that with variables instead of having the whole words. And basically, this adds up to the growth rate of the population. And we're going to be referring to growth rate as R, so just try to keep that in mind. That's how much the population is growing. You actually saw that a little bit last unit also. So we've talked about limiting factors already, and remember, limiting factors are anything that limits the size of a population, so it's going to determine whether individuals live or die. So 2 types of limiting factors. Density independent limiting factors impact the entire population equally. So these are completely random. They're not picking one individual over the other. These are things like natural disasters that would impact everybody equally. Density-dependent factors, on the other hand, impact certain individuals more than others. So this would be anything that causes competition or natural selection in a community. That would be density-dependent. So some examples of both of these are here, and you can see that all of these things are pretty much random, and it's not picking out certain victims. Now, one exception to that, pollution can be density-dependent. Sometimes because it's tied, resistance to pollution is tied to genetic makeup. So that could be considered density dependent. All of these density dependent factors usually depend on some form of competition. So as your population size increases, it's going to be harder for them to find food, mates. It's going to be easier for the predators to get them and sorts of things like that. So that's why we call them density dependent. They depend on how many organisms you have. So again, these limiting factors are going to basically limit your population growth. And no population can just keep growing forever. So we have 2 different variables that we're going to look at here. R, which we've introduced you to already, is the growth rate, or another term for it is biotic potential. Basically, how fast the population is going to grow if it has unlimited resources. So this is known as exponential growth. They're all kind of the same thing. Biotic potential, R, exponential growth, growth rate. All right, and then our other factor we've also talked about already, which is carrying capacity, also called environmental resistance, or the letter K is used to stand for this. And this is the maximum population that a particular habitat can support, sustainably anyway. So this is logistic growth in comparison to exponential growth. So if you remember when we talked about our selected species, we said they're just going to grow and grow and grow and try to reproduce and reproduce. Well, that goes along with this exponential growth curve. You get this J-shaped curve, and it is basically just going up unchecked. So bacteria are the easiest examples of this. A lot of times, especially if they're resistant to drugs, you will see this growth rate in them where it's just insane. Human population is currently following an exponential growth pattern as well. And when you look at those graphs, you'll see this J-shaped curve. All right, so there is a way to calculate this, and I'd like you to pause here and record this. All right, so basically, if you want to know the population at a given point in time, you have to take the starting population and multiply it by, in parentheses here, 1 plus r, that's the growth rate. So you take 1 plus r, you put that to the power of t, which is the amount of time you're interested in. And then once you calculate that, again, you're multiplying that by your starting population. And that will give you your population at that point in time. And again, that's depending on how much time has gone by. Now, logistic growth rate is when we hit that carrying capacity. So once you hit that, your J-shaped curve turns into an S-shaped curve. So remember, those K-selected species are in it for the long haul. They're going to stay closer to that carrying capacity. That's why we call them that, because they're at K there. So typically, populations start off in a J-shaped curve. They start off with exponential growth, and then over time, they reach the limits of their environment. So they reach that carrying capacity, and then they start to just fluctuate very close to carrying capacity. So as the populations are getting closer to K, carrying capacity, the density-dependent factors have an increasingly negative impact. So anything that they're competing for, the competition's going to get harder. And that's going to slow their growth rate. So that slows R down, and it decreases R growth rate. If the populations decline even further to below K, at that point there's going to be some relief from competition. So at that point R would go up, and you would start to see the population grow again until it hit K, carrying capacity once again. But you do often see this continuous fluctuation around carrying capacity where the population will overshoot it, die back off, come back up again, and just keep going back and forth around this. So we're going to talk about some population patterns coming up here in the future. Right. Now, that's basically what has to happen. Either you have that overshoot and it just keeps going back and forth like that, or the other option is the animals have to emigrate. They have to leave and find another habitat, or you get a population crash. And in some cases, it can be significant enough if the environment has degraded enough and the carrying capacity has lowered enough, the entire population could wind up going extinct. Alright, so we're going to talk about all of these factors in the coming days and these different patterns of growth and populations. And just make sure that you come in knowing what all of these things are and you ask questions if you're not sure about any of it. Alright, thanks, and I will see you after break. Enjoy.